In laboratories across the world, the frontier of genetic engineering is being redefined each day. Yet few recent developments have sparked as much intrigue and controversy as the groundbreaking work of Chinese researchers who are blending the boundaries of life itself. Their focus lies on a creature so small it's almost invisible, yet so resilient it has earned the nickname the indestructible animal. Tardigrades, or water bears, have long fascinated scientists for their uncanny ability to survive conditions that would annihilate nearly any other form of life. These microscopic organisms can withstand the vacuum of space, absorb doses of radiation that would be fatal to humans, and even enter a suspended state to survive years without water. But the latest twist in this remarkable story goes far beyond the study of these creatures in isolation. Chinese researchers have begun using CRISPR, the revolutionary gene-editing tool, to merge tardigrade DNA with human stem cells. Their goal is both ambitious and controversial: to confer some of the tardigrade's legendary resilience to human cells, potentially allowing them to survive radiation, grow faster, and perhaps one day endure otherwise lethal environments. At the heart of this research is a unique protein found in tardigrades, which shields their DNA from damage caused by radiation and other extreme conditions. By introducing the genes responsible for this protein into human stem cells, scientists hope to create cells that are significantly more robust than their natural counterparts. Early experiments have shown promise, with modified cells displaying greater resistance to X-rays and enhanced growth rates. Such advances could revolutionize medicine, offering new hope for patients undergoing radiation therapy and opening doors to treatments that were once thought impossible. The medical potential is vast. Imagine cancer treatments where healthy cells could be shielded from the collateral damage of radiation. Consider therapies for genetic disorders that currently leave cells vulnerable to environmental stressors. In the realm of regenerative medicine, more resilient stem cells could mean faster recovery for patients with debilitating injuries or diseases. Even beyond hospitals and clinics, these breakthroughs could impact people working in hazardous environments, offering protection to nuclear plant workers, astronauts, and emergency responders exposed to radiation or other dangers. Yet, the implications extend even further. In the sphere of military research, the prospect of soldiers with enhanced resistance to radiation is an enticing one. In the grim scenario of nuclear conflict or radiological accidents, such abilities could mean the difference between life and death. The idea of creating military personnel with biologically enhanced traits is no longer confined to the realm of science fiction. It is becoming a real possibility with all the promise and peril it entails. But as with any leap forward in biotechnology, these advancements raise profound ethical questions. The integration of foreign, non-human DNA into the human genome challenges our most basic understandings of what it means to be human. Are we crossing a boundary that should remain inviolate? Or does the pursuit of knowledge and human flourishing justify such experiments? Bioethicists warn of a slippery slope. The line between therapeutic intervention and human enhancement is thin, and the temptation to use these technologies for purposes beyond medicine is strong. If we can make cells more resistant to radiation, what's to stop us from seeking enhancements for strength, intelligence, or longevity? The prospect of designer humans, engineered for specific traits, raises concerns about inequality, social justice, and the very nature of humanity. Moreover, the long-term consequences of such genetic modifications are unknown. While CRISPR allows for precise editing, the introduction of foreign DNA could have unintended effects, both for individuals and for future generations. Mutations, immune reactions, or unforeseen health problems could arise, echoing down the generations in ways we cannot yet predict. Internationally, the debate over genetic engineering is intensifying. While some nations have enacted strict regulations on human gene editing, BOSI, others have pushed ahead, driven by the promise of scientific leadership and technological supremacy. China's willingness to explore the outer boundaries of biotechnology has drawn both admiration for its boldness and concern for its ethical oversight. The global scientific community is watching closely, calling for transparency, collaboration, and a robust ethical framework to guide this research. Public opinion is divided. Some see these advancements as the next logical step in the evolution of medicine, a necessary pursuit in the face of disease, disaster, and the unknown challenges of the future. Others fear the loss of our shared humanity, the commodification of life, and the potential for abuse by governments or corporations seeking advantage at any cost. To navigate these uncharted waters, open dialogue is essential. Scientists, ethicists, policymakers, and the public must engage in honest conversation about the risks, benefits, and values at stake. What kind of world do we want to create? What limits, if any, should we place on our ability to rewrite the code of life? As research continues, the need for clear regulations and ethical oversight becomes ever more urgent. International organizations are working to establish guidelines, but enforcement remains a challenge in a world where technology moves faster than policy. The questions raised by the merging of tardigrade and human DNA are not just scientific. They are deeply human, touching on our hopes, fears, and aspirations for the future. In the coming years, the results of these experiments may reshape the landscape of medicine, defense, and even what it means to be human. The promise is extraordinary. The ability to heal, protect, and enhance human life in ways previously unimaginable. But with great power comes great responsibility. The choices we make today will echo through generations, shaping the destiny of our species. In the end, the story of CRISPR, tardigrades, and human enhancement is not just a tale of scientific ambition. It is a reflection of our collective journey. A journey defined by curiosity, creativity, and the relentless pursuit of progress, tempered always by the wisdom and humility that come from understanding both the limits and the possibilities of our power. As we stand at this crossroads, the future remains unwritten. It will be shaped not only by the breakthroughs of researchers in distant laboratories, but by the conversations we have, the values we uphold, and the vision we share for the world we wish to build. In the quest to unlock the secrets of life itself, the greatest challenge may not be what we can do, but what we should do, and how we choose to define the meaning of being human in an age of limitless possibility.